go to school, did you remember that game that they played for converse at one time?
Oh, that's
ours was a cone, but it was the black circle cone that had the hole in the middle. So I want to grab it from the middle, pull it towards me, and pull me on that asphalt. Wow, my whole nail. Like my nail was literally gone.
Y'all play?
We played at workout. But we all went to workout this morning. And Jocelyn led us to. And Savannah left and Shelly. There's 7 Eric's with them.
So then it's like, so we're going to work out in the morning and we're good. You guys know we're going 4:30 a.m. after that. I'm saying it's 2:30 last night. Yeah, so you know they're going to be up for
Good morning. Can you see it?
Yes, sir. Morning.
Morning. Okay, we just start. So as you can see, chapter 3 to 5. So it basically just have a quite similar content. So it's just about the cell structure of bacteria, archaea, and eukaryotes. So here just combined together, easy to simplify. So then easy to talk about it. So I think most of you know it because you guys took the gene biology lab 1 and 2, right? So now, so for this content, we probably can introduce a little bit fast. So if you have any questions, just let me know. Okay. So first, we talk about the bacteria cell structure. So you can see this. Always we compare with like a eukaryotic cells, right? So when we talk about like a bacteria archaea, then you can see the major difference is they don't have like a bacteria archaea don't have like a membrane bound nucleus and organelle. So that's a major difference. Then the so-called prokaryotes sometimes as blurs. It just refers some like prokaryotes like a cyanobacteria, right? So yeah, microbiologists consider as like a bacteria because they don't have membrane-bound nucleus. But botanists, they consider as a plant, right? Because they also contain like pigments. They can do photosynthesis. So that's basically what about this kind of sentence here. Then you can see the general fish, like a morphology for microbials only have like a 3 to 5. So here about 3 to 6, so different like shapes. So the major like 3 morphologies are the cockles. So we know like a small bean, like tiny. Then rod, just like a kind of stick. Then spiralium, so you can see the kind of curve kind of structure. You know, vibrant, right? So that's it, like a kind of typical kind of morphology. They also have some not very common like a morphology, like a spiral key. So it's just a little bit longer. Hyphae, so or hypher or stalk, so like that. Then some bacteria, like a cyanobacteria, they have filament kind of structure. So in the summertime, when you if you have time, you can take some water. So from like the university lake, so then you will see actually, especially in the like there's a lot like algae bloom, and you see a lot of kind of filament bacteria or algae. So see, this is just a look unclear like a kind of classification. Then also some microbia, they can have a kind of so-called arrangement. So just several cells, they stick together so that we form a little bit unique kind of morphology. So like so-called cocci, so just several coccus kind of cells, they stick together. So like as a pair called diplococci. So we have a 2C here, pronoun as I here, cocci. So then streptococci, it just they arrange as a kind of chains. So like they here. We're not streptococcus, right? Kind of bacteria. We probably will use in the lab. Then staphylococci is just like a grape. So they have a kind of clusters. So also we know like as Staphylococcus aureus, right? Aureus. So this is just a kind of very typical species pathogen. And also we stick as a 4 or 8, then they have a different name like a tetride, so saccinate, that's just a kind of name for those kind of arrangement. Then also have other like bacilli. So we know this is just a bacillus, right? So it's a kind of bacteria, bacillus. So that's where the name comes from. Okay, bacillus, we know Vibrio. So that's the same. So that's also this in terms of morphology. So we know there's a lot of kind of marine or coast region, those kind of pathogen called Vibrio, right? It causes diarrhea. We talk about later. Spirilla, so just spirillum, so spirochetes. So that basically see that this is just a kind of different morphology. So then you can see, oh, we only have like a 3 to 6 different morphology, right? Can we distinguish bacteria or archaea as just based on their like a kind of morphology? It definitely is not, right? Because we have like a thousands, even the millions of different species right now, just waiting us to discover. So, so you can see that's why last class we introduced so-called evolutionary ruler, right? So like this, the 16 sRNA genes. That's actually a really fundamental, like the basis to distinguish the millions different species. So not like the animals or plants, right? We are actually based on their morphology to distinguish different species. So then you can see sometimes being small, actually we okay, this is actually the size. Okay, then I really hope we can at least remember those kind of average size for them the E. coli. So this is a kind of typical size. You can see the cells, actually there's 2 ways, right? 2 dimensions. One is the length, another one the width. So wide, long, right? Or the long actually is so-called the diameter. Can you hear me? It shows some like unstable internet. It's okay, right?
You cut out for a second, but
Oh, okay.
I can hear you.
Thank you. So then E. coli, you can see the usually we talk about the size is a diameter, okay, the length. So how long it is. So then it's 2 to 6 micrometer. So that's the kind of size at least you would know because we use E. coli very often, right? So this is the kind of typical size, like 2 to 6 micrometer long. Then what show here is a nanometer, right? Nanometer actually is 1,000, like smaller compared to micrometer. So that's why here you can see like this. The length is about 4,000 or 4 micrometer. Okay, 1.3 micrometer. This size. Okay. Then you can see I also have a different size for for microbials. Some like this is oxalatoria, so it's essentially kind of cyanobacteria. You can see they're quite large, right? Then some streptococcus is a little bit small, like just 1 micrometer. Then if you compare with the size of a virus, then you can see that here, this is several pox virus. So then influenza virus, then E. coli, T4, like bacterial phage. When we talk about phage, means that they actually live in, so like a T4, they live in E. coli, feed on it. So you call phage. So usually that means that it's virus. Tobacco mosaic virus, then especially, so you can see this one. So polymyelitis virus, you can see very small, right? Compared with E. coli, just like one person stand in a kind of stadium, right? So that's why a lot of kind of virus, they can live in within the bacteria and feed on them. So this is just like kind of relevant. So you know the relative size. And some bacteria is very large. So this is actually a kind of bacteria, original isolate, as in one kind of fish. Then you can still have the size diameter is 80 micrometer compared with the common, so 2 to 6. Then you see the small size actually is good for microbes. So then here, just give an example. Then when like the radius, so which is like a 2 millimeter, right? If you reduce to like 1 millimeter, then we can calculate the ratio of surface versus volume. So you can see that actually increase the double, right? Double kind of ratio. So this ratio actually is really important because specific like a surface, right? So if it's bigger, that means they have a large interaction surface with the environment. So which is good for them to take a nutrient. They have more chance to interact with surrounding like water if they stay in like a lake, like pond or river. So then here's just a kind of general feature for bacteria. And when they always we know that they have 3 layers, like for cells, so-called envelope. So always have in the inner part, so plasma membrane, then cell wall, then outside. Okay, I have a chatting. Let me see.
You just cut out a couple times.
Oh, I cut a couple times. Okay, that's good. So it's better right now.
Right now, yes.
Okay, I'll talk a bit slowly. Okay, thank you for the kind of message here. Okay. Then you can see typically have 3 different layers. Okay, then within the plasma membrane, we know there's cytoplasm, right? And this is actually kind of the P. So this we call it external structure. It just
Sir did it again.
Oh, did it again? Oh, okay, I just need it. Then you can see yeah. Okay. Because of my rollers, they are away from this room. Okay, we will talk about lab. Okay, don't worry. We will have online, okay, the Zoom meeting, same, because as the university policy, we cannot go to lab this week, right? So we will have a Zoom meeting. So I have the website. I will probably send you guys again. Don't worry about it. Anyway, I talk about, just let me know, okay? I will talk a little bit slow. So then Then you can see this is just a kind of online structure I found in cartoon. So it just indicated the general kind of structures on microbial cells. So then you can see they actually contain a lot of kind of small organelle, right? So like the ribosomes, you can see they have a lot of ribosomes, right? They actually need it. So when they wanted to grow, right, they need a lot of protein, synthesize a lot of protein. Then nucleus or nucleolide. So basically, that's not membrane, right? Then you can see a list. This table lists kind of function for the different organelles on like all structures on like a bacteria, like a general structure. Okay, then we talk about more detail about like the plasma membranes. So then here, I think this is the better one. So you can see really clear, right? The 3 layers for so-called cell envelope. So so-called envelope. So that basically just include like the inner layer, which is a cell membrane, right? Or plasma membrane. Then in the between is cell wall. Then outside, it really depends what kind of bacteria. So like a gram negative, you probably don't have a lot, but have really thick cell wall. Gram positive, they have a very complicated, or the so-called external or outer structure. Basically, like just a kind of captures slime layer. Okay. Then you can see again, you can see the nucleolide, right, which is basically just contain all the chromosome DNA. And then again, you can have a lot of kind of ribosomes, fragile. Okay, then this is called fibria or pilus, right? So anyway, we talk about it later. So when we talk about conjugation, actually now some like the fibria actually is very important. Then the function we all know. So this is the kind of layer, right, to separate the cytoplasm, include organelle from the outside environment. But the actual content, they have even more important other like functions, like as a kind of barrier, which actually can allow the nutrient to go inside and waste to go outside, right? It's so-called permeable barrier. And they also contain a lot of protein, so which can act like receptors, transmit nutrients or waste, and do a lot of kind of metabolic kind of processes. Then you can see this is just a kind of basically structures. So always we know the cell membrane actually usually contain 2 layers, right? The lipid structure, it's so-called lipid bilayer. So then you can see there actually for each layer, they have 2 ends. So one is so-called polar end. So that actually is usually just like outside or this inside, right? You think about this is cell. So this actually is inside, like inner, this inside, face outside. So then another 2 ends. So or just so for one layer, this another end is so-called the non-polar tails. So basically, the so-called hydrophobic, they are actually phobic is afraid of. So it won't be like a soluble. It's not. It doesn't like water. So this end actually very inside. Okay. Then another end actually they can interact or soluble with water. So this actually the structure is very important. So that's why the this kind of membrane they can act like a permeable barrier. Then in the between, you can see embed a lot of kind of protein here. So so-called membrane proteins. Usually have 2 kinds. So you can see if it's on the outside, so-called the peripheral, so like that protein. If it's gonna embed in the membrane, so which is called integral. Integral is kind of blue one, okay, protein. So this kind of protein actually can act like a so-called channel. We talk about a channel. They actually say it can allow some nutrient pathway, so which is really important. Then you can see peripheral protein actually attached to either membrane, right, or the protein. So then also have other structure you can see here. So the membrane actually is quite complicated. Then for the structure of lipids, so we talk about the bilayer, right? The lipid bilayers. So what kind of structure? I mean, consistent of them. So that's basically about. So like you see, there are just a kind of difference between like nuclear outer cells membranes. So which are just usually is consisted of like a cholesterol. We know this is actually in terms of like a health. So it's not good, right? But it's very important material to make the membrane, so the cell membrane. So look at the structure actually a little bit different, right? You have a orange here, but the lipids for bacteria, so then you see that actually is different. So I have a 5 range. So the name is different, so called haploids. So then versus the cholesterol, so which is like that. So there's you can see the structure is a little bit different. So I hope at least you know, like the chemical names. So between this eukaryotic cells and the bacteria. Then we talk about lipid nutrients. So we know the so-called the micronutrients. Right? Micronutrients is just that they need a lot, so like carbon, nitrogen, phosphate. We know, right? All for plants, right? Human, bacteria, the same. So that called micronutrients. Also some like ions, so potential calcium, magnesium, iron, like they actually needed this kind of a lot amount. Then some elements actually they need a very small amount called trace elements, okay. Like zinc, copper. So no, we toxic, but I still need it. Like molybdenate, so nickel. Then the reason they need it actually they can use it for like an enzyme, so-called cofactors, especially iron. Right? We know they need iron, so that like human, we need iron too because the red cells, right? They need it. Then also have like a growth factors. They need a growth factors. So the reason they need it is because they cannot synthesize. So like amino acids, vitamins, bases. So and here just give more details. So like some amino acids bacteria actually cannot synthesize. Purines, pyrimidines. So that's for nucleic acid, DNA, RNA synthesize. Right? Vitamins, hemes. Again, you can see the iron actually is very important. It is same for microbials. And then we talk about a little bit the uptake mechanism for nutrients. So this is actually a kind of basic physiology process, right? We all know what happens in nature when you drop like a little bit sugars or salt right, in the water that we can like diffuse right away, right, to reach to equilibrium. So this is the kind of phenomenon we call passive diffusion. Okay, always just happen by nature, move from like a high temperature, high concentration or lower. So that called passive diffusion. They also have others, so like a facilitated diffusion, okay, active transport and group translocation, Then for eukaryotic cells have endocytosis, right? So that's a little bit different. First, we'll talk about passive diffusion. So this actually happens by nature. So always like nature determined, right? So if I have high concentration, they will definitely can be diffused right away. So the reason we call this by nature is just they don't need energy or protein to help. So then you can see here, just have a video you can take a look.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Okay, then another kind of diffusion mechanism is called facilitated diffusion. You can see quite similar. It doesn't need an energy. Then like a diffuse from high to low concentration. But there's a little bit different. You can see they actually need a protein to help. We talk about a protein embed on the cell membrane, right? That's kind of the so-called carrier proteins. So they can open it, so let a nutrient or waste go in or out. So that's a so-called channel, okay, or carrier proteins, basically the same. Then you can see here, this figure actually compare the differences between these two. And if the chemical is diffused by passive diffusion, then you can see once the concentration increase, then you can see the rate of transport will increase linearly. So that's a so-called passive diffusion by nature, right? But then the facilitated diffusion actually is a little bit different because the cells need a protein to help, right? So think about it like E. coli in the lake water, right? They need to take the nutrients. Then you can see they actually say once they have a nutrient, even at very low concentration, you can take up very quickly. So you can see the initial, the rate, so-called rate transport, it means the speed, right? You can take it how fast you can take the nutrients. Then even very low concentration, you can see they take it very fast. Then, but we reach some saturation plateau. So the so-called saturation. So this is actually a little bit different. And think about it, this is just a way for microbia to survive, right? In the natural environment, once they find that there's nutrient around in the environment, they will definitely, try to take the nutrient as soon as possible, but they need the nutrients. So that's a difference, okay? They call facilitated diffusion. You need a channel or protein or permeases to help for this kind of whole process. But it does need a new energy. So this actually basically the same. Then also a transfer from high to low concentration. Then another one is a so-called active transport. Okay, this is the so-called active transport is just like the bacteria, they take nutrients or chemicals, which actually is against the gradients, the concentration gradient, from low concentration to higher. So that's a so-called active transport. So then you can see this actually need energy, right? Either it's ATP or protein molecule force. So we'll talk about later on. Then they need protein or carry protein or permeases to help this process. And there's three types. Okay, primary active transport, secondary active transport, and group translocation. And the first one is a so-called primary active transport. So it need like a specific protein, ATP binding cassette transporters or ABC transporter. So this is just a kind of protein, okay, several protein, so-called transporters. Then you can see this is very popular. So you can see bacteria, archaea, eukaryotes. And basically you can see this ABC transporter includes three parts. And you can see the first part we know the embed protein, right? This is so-called the transporter. So the membrane spanning domains. So they span across the membrane. So then you can see the protein actually they can open, right? Then as a channel. So that's why we call it channel. And the second one is they see some protein attached with this kind of protein, right? So that's called cytoplasmic associated ATP binding domains. So the reason we call that is because this kind of protein that we bind ATP, so we use ATP to get energy, so help the transport the nutrients or waste. So then you can see the ATP will attach on it, then dissociate, right? And that's the way to give energy, right? So then you can take up the nutrient. Then the last part is so-called the binding domains or binding protein. So that's a kind of protein bind on the substrate and carry to this kind of channel. So then you see this is so-called ABC transporter. That's quite interesting, right? They actually can work together so to carry out the nutrients they need. And then secondary active transport, you can see these are a little bit different. So they actually not directly like use energy. So they actually couple with those kind of the ions. So especially sodium, proton. So sometimes you see once the cells do some process, they actually we create a kind of gradient, right? So like the protein will move out to the outside the cell membrane. So they can use that kind of like a so-called protein molecule force, right? So so-called ion copper transport. So once they move in, they can carry the nutrients in, so that or they can opposite way. So it really depends. So then that's so-called the ion copper transport. So they basically use iron as a kind of energy source to do the nutrient uptake. So there's a different way. You can see the so-called simple. So it's just the same way. So like the iron, you can carry in also the bring the nutrient. Okay, this is a part nutrient. Okay, then another so-called the antipode, so upset. So when like the sodium or protein move in, they actually can create a kind of force to move out the waste, okay, for example. So that's actually something the cell needed to get rid of, right? Or just a so-called one-way unipolar.
Small
So then you can
molecules such
see
as
the video probably not better.
Small molecules, such as sugars and amino acids, can be transported up a concentration gradient. The sugar moves via a membrane transport protein from outside of the cell where the sugar concentration is low to the inside of the cell where the sugar concentration is high. The transport of the sugar through a coupled transport protein is driven by the movement of counterions such as sodium ions or protons that are moving down their concentration gradients from a region of high to low concentration. Sodium ions and the specific sugar or amino acid simultaneously bind to the same transmembrane protein on the outside of the cell, called a symport. When the counterion is sodium, the low concentration of sodium on the inside of the cell required to transport the sugar is maintained by the sodium-potassium pump, which is powered by ATP. In a related process called countertransport, the inward movement of sodium ions is coupled with the outer movement of another substance such as calcium ions. As in cotransport, the sodium ions and the other substance bind to the same transport protein called the antiport, but in this case they bind on opposite sides of the membrane and are moved in opposite directions. The low internal sodium ion concentration is maintained by the sodium-potassium pump. Group translocation.
Okay, this is just another kind called the group translocation. You can take a look, video first, and then we talk about it later.
Group translocation is a process in which an organic molecule, such as glucose, is transported into the cell while being chemically modified. The PEP phosphotransferase system is an example of group translocation. In this process, the phosphate from the high-energy compound phosphoenolpyruvate, or PEP, is transferred to HPR by enzyme 1, and the product pyruvate is released. The phosphate is then transferred from HPR to enzyme 2B by enzyme 2A. The membrane-bound enzyme 2C then carries the glucose across the membrane, and the glucose becomes phosphorylated as it enters the cell.
Okay, then you can see this kind of so-called group translocation systems. Just first, they involve several enzymes, right? That's probably what I call group translocation. And then another feature is just a so-called chemically modified. So once you take in like a glucose, right, you add a phosphate. So when we talk about glycolysis, then for those of you if you take a biochemistry, you know, right, this is just a very important step for glycolysis. Then this is another feature. They modify, so-called modify. They add a phosphate on glucose, so that's modified. So anyway, they involve several like protein enzymes here, so to take in glucose. So that's basically two features. Don't worry about all the details. Then the use kind of chemical is actually not ATP, right? They use so-called phosphoenolpyruvate, or PEP. So this actually is also very important, like intermediate products. We talk about glycolysis, right? And also you can see this actually contains a lot of energy. So it just acted like an ADP. So they can use other like intermediate products as a power source to moving like nutrients. So iron actually always is a kind of quite difficult for for a lot of organisms to take up, right? So the reason for that is because it's the ferric iron, iron 3 actually is not easy dissolved in water. So then that actually happens in algae, right, in ocean. Now so-called iron limitation. We take like oceanography, especially in those kind of deep ocean. So actually there's very few algae. So the reason for that is just it's short of like an iron. So algae cannot grow very well. then it happened to bacterial archaea too. So then how can they deal with that kind of situation? They actually, especially those microbes, think about it in soil, right? If the soil doesn't contain a lot of like iron, so that's a kind of problem. And they have a developed kind of system. So basically just a kind of protein peptides called cytophones, structure like that. So they actually can bind on ferric iron. So make the iron and the whole like a band complex just soluble. So then bacteria can easily take it out. So at least I hope you know what side flow refers. Okay, that's why I draw a line here. So anyway, so this is just a kind of so-called ion carrier. You can bind on iron, so then make it easily, kind of available. Then we talk about more about the cell wall, okay, bacterial cell wall. So this is actually very critical, so especially for bacteria. So then you see this so-called the peptidoglycan, peptidol, right? Peptide. So basically it's a piece of amino acids. So or protein. Glycan is sugar. They have another name called murin. Okay. So then this is actually very unique for bacteria. So we know archaea, eukaryotic cells actually don't contain this kind of a structure or molecules. They actually have another kind of structure. So that this is actually very unique for bacteria. And then this is also the basis for a very classic kind of assay called ground staining, right? We will learn in the lab. So then the ground staining actually is really use a kind of dye called crystal violet to stain cell wall. Basically is this kind of structure, peptidoglycan. So when the cells or group of bacteria which contain thicker peptidoglycan, then they will show kind of very unique color, purple, right? So that's basically what about it. So you can see if it's gram positive, so the cell membrane which we stain as purple. And if the bacteria only contain like a very thick, very thin peptidoglycan layer, it will turns purple. It turns pink or red. So that's so-called gram negative. So basically, like this kind of bacteria, like E. coli. So that's why we can classify the bacteria, all bacteria, into like two kinds. Right? One is gram negative, one is gram positive. Okay, then here just give more features. You can see the gram positive bacteria, like a bacillus. Okay, they have a quite thick, like the peptidoglycan. So on the cell wall. Okay. Then for like E. coli, so they only have a very thin layer of peptidoglycan. So then once we add the dye, so a crystal violet, so then you can see we take up some this will take more, right? The dye, right? And take less. Then we add like ethanol or those kind of decolorize, then we cause a shrink, right? Then for those cells with thick, like a peptidoglycan, they will hold more dye. So then even you remove the color, so they still can remain the original color, like a color of dye, which is purple. But for those have a thin layer of peptidoglycan, they actually cannot hold a lot of dye. Will be removed by like those kind of decolorize agent. So that's why they can turn like a pink or red. So that's the way actually. Then you can see this is what it really look like under like a electron microscope. So you can see peptidoglycan for a positive, gram positive of a thick layer, and then that's now also not much outside in kind of structure, right? For gram-negative, you can see there's only just a kind of tiny here. This is a tiny one, but they have a outer membrane, right? So, then this is a kind of typical response, and you have to remember. So, like a gram-positive, purple, gram-negative have a red after staining, gram staining. So because you probably will face this kind of exam all over. So a lot of kind of like MCAT or something. So the way I'm remember actually pretty easy. For them, this is the so-called gram positive, right? It's positive. So that means they have a thicker peptide glycan. And then because it can hold, because it's thicker, it can hold more dye. So then they have the color like a purple color, which is the original color of crystal violet, so-called violet purple. Then negative, so then that means they have less peptide glycan and red. Okay, I hope that will help. So you'll probably find a better way. So anyway, find a way to remember it. Then the function actually is quite obvious, right? The actually are very solid. You can keep the kind of morphology, the shape of the cells, and protect the cells from like toxic pathogens, osmotic lysis. Then here just give more detail about the structure of peptide glycan. So that probably help you know better. So this kind of structure, you can see the peptide glycan actually contains like 3 typical kind of parts. So then the first part is so-called the nitrogen acetyl glucosamine, or NAG. So then you can see here just divide these 3 parts. Then you can see we can talk about one by one. We know acetyl, right? Acetyl means you have 2 carbon right here, carboxyl group. So that means this part. Then amine is actually the nitrogen and hydrogen, right? This amine group. Then they have a glucose here. So that basically just says so-called acetyl, glucose, amine. Okay, that's basically this part. Okay, then another one is that nitrogen acetyluronic acid. So that's the same. You can see you have a acetyl here, right? See, it's 2 carbon carboxyl. Then they have sugar. Okay, then they have actual one is lactic acid. So they have here. So we know lactic have 3 carbon, right? Carboxyl. So that's basically these are 2 major kind of structure. So called NAM. Okay, NAM here. So the NAG, NAM. They also have another one so-called side chain. So basically amino acid side chain. So then you can see how about alanine, right? Alanine, I'm sorry. And glutamic acids, and also other like alanine. Anyway, basically just a kind of amino acids side chain. You have 3 parts. So this is just a basic structure. When we talk about DNA, RNA, we know the basic structure, right? And phosphate, like, sugars, and base. So this is the basic thing. So we have 3 parts. Okay, NAG, NAM, and side chain. Then the basic structure, then once we combine together, then forms the whole, like a structure, like the DNA structure. Okay, helical kind of structure. You can see they have a NAG, NAM, then peptide side chain. And it's the same, so you can see they repeat over and over and over. So then finally can form a structure like that. So just like a membrane. Okay, the cell wall, right? We know the cell wall. So sometimes you can get it, once the cell died, you release all those kind of cytoplasm and nuclear. Only remain cell wall. So this is what it really look like. So about like a 0.25 micrometer. Okay, this is for bacillus. We know bacillus is positive, right? Gram-positive, they have thicker peptide glycan. So once you know this kind of structure, then you're more familiar, right? So You can see they are sometimes they have even more complicated kind of structure, so-called the interbridge. So not only have the three like a side chain, there's two, right? They also have a kind of interbridge to connect the two like units. So this is for this is from Staphylococcus or E. coli. Then here just to talk about a little bit more about the difference between like gram-positive and gram-negative. So we already know, right? The gram-positive have thicker peptide glycan. So and then this is what it really look like. So you can see this is M stands for murine. Okay, so that's the kind of layer. We know gram-positive bacteria actually they don't have a lot outside kind of structure, external, right? So that's also basically just the peptide glycan or cell wall. Then this is the plasma membrane. So you can see this this kind of color. Then sometimes they also have other structure like tachoric acids. So that's just kind of help them to make it more solid. So like this. Okay. So just stick inside the membrane, so make it more solid. Also have a kind of connection with this the plasma membrane. So then, oh, this is just a kind of comparison between gram-positive and negative. You can see gram-negative actually have more the so-called periplasma space. So that's basically kind of space between plasma membrane, right? Or cell membrane and the cell wall or peptide glycan layer. So you can see you have a more space for gram-negative compared with gram-positive. Anyway, this is just a kind of tiny difference. I really hope we can know what's the difference between these two. So in that way, we know better. We know all, like most of the pathogens are gram-negative, right? So it's just, now you can see the space actually is not 100% useless. They actually contain a lot of kind of enzyme. Then here you can see the, just give an example for the, like the space. So you can see the gram-negative have a lot of space here. So sometimes I have the space occupied a larger kind of area. And this is just a kind of cartoon. You can see much better. So for gram-negative, so have a very thin like a layer of peptide, right? Glycan, but the space actually is quite large between like the plasma membrane and the cell wall. Then this is the outside membrane, right? Especially for gram-negative. There's no tachoric acid. Okay. Then for the external or outer membrane, we know the major component is so-called the lipopolysaccharide or LPS. Okay, I hope we know what LPS stands for. Okay, it's just a lipopolysaccharide. We know lipo is lipid, right? Just a kind of fat plus some poly sugars, right? Polysaccharide is just a kind of sugar. Not only that, this is the major one. So those are kind of slime, so sticky stuff. So basically just a kind of lipopolysaccharide. Then the plus some like a lipoprotein. So then you can see this actually is a kind of lipid plus protein, right? It's a kind of mixture. Then lipids. So that's basically about the outer membrane for gram-negative. We know gram-negative bacteria, they have very complicated outside membrane, right? So they not only they have like a peptide glycan as a cell wall, they also have outer membrane. So for gram-negative, you can see when we talk about a cell wall, they actually contain two parts, right? So one is outer membrane, or LPS dominate kind of structure, plus the traditional way, the kind of cell wall, so peptide glycan. Anyway, here's just a list like the percentage. Okay, peptide glycan, you can see for gram-negative is only represent 5 to 10%, but the space actually is very large, occupy like 20 to 40% of cell volume.
Professor, you got a question in the comments.
I have a what?
A question in the comments.
Oh, I have a question. Okay. Let's see. You mean in the
It's in the comments, in the comment section.
I don't understand what I'm saying. In the comment section?
In the chat.
Oh, in the chat. Okay. Let me see. Oh, here. Okay. Oh, okay. Good to know. Anita, okay. Is the old membranes of gram-negative cells considered as part of? Yeah, this is actually part of the cells. That's a good question. That's actually kind of captures a slide. Yeah. So that's what I'm saying here. See, this actually it have a indicate it indicated here, right? Cell wall. The so-called cell wall for gram-negative actually contain two parts. So outer membrane or the slime, or the outer membrane plus peptide glycan. Okay, that's what I'm saying. So that actually, so when we talk about a gram-negative, actually it contain two parts, but gram-positive actually they have a very few outer membranes, right? So that basically just you can see the for gram-positive actually the only. I'm not sure there's few bacteria probably have liver, but usually only have like a peptide glycan for gram-positive. So when we talk about the cell wall for gram-positive, so basically probably it's just a peptide glycan, like a bacillus. Okay, thank you for reminding me. Okay, if you cannot hear you, just make it, you can unmute, then talk to me directly. Okay, that's probably easy way to communicate. Because I cannot see you. Now see you to raise a hand, something. So we just make it some noise. So I don't mind for that. Okay, that's the way I mean. Better to have better communication, right? Okay, then we talk about the more about the the structure. It actually is a little bit more complicated for gram-negative. So then you can see here, this I give more like a details of like a the cell membrane, right? So or plasma membrane. So we talk about like there's a lot of protein embedded, right? You have a kind of different transport mechanism, which is talk about. Now also they have like a peptide glycan. Okay, this is the peptide glycan. So the yellow one. Then also they have talk about a cell wall. They have an old membrane. Okay, so on old membrane actually you can see not only like those kind of like a LPS. They also have protein. You can see like the porin protein. So LPS is here. You can see this is kind of like the fragile like the structure. So lipopolysaccharide. So this actually is really important for some bacteria. They cause like a disease. They kind of antigens, so acting like antigen. Then you can see the protein here actually have functions. It can help the cell to communicate with outside, transmit some signals. For example, they can have like a chemical signals pass out or pass in. So they have a kind of link between the three parts. But the two parts we consider as a cell wall, okay, old membrane plus the peptide glycan. So then this is inner or plasma membrane. Then for LPS, you can see just this part, okay. It's a kind of like a fragile like structure. Then you can see they usually have three parts. The base part is called oocyte. This is as a lipid A, I'm sorry. So this is the kind of base, so it can embed on the cell wall. Then also have the core polysaccharides, and plus the O chains or O antigens. So act as a kind of antigen, well, not antigens, right? That's the way to cause a kind of disease. Anyway, that's LPS. Then here just mention the role of LPS. We know they have different roles, right? So then is one way is they can help like bacteria to form like a biofilm or attach to surface. So that's probably first like a human hand, right? They can touch somewhere to pull and then stick on the solid phase. And also they can help for the nutrient like transmission. So permeability as a kind of permeability barrier. Oh, that's actually thinking block some way, right? Block some like toxic stuff or particles. Also can act like antigens or toxins. So let me say some bacteria, the reason they are really toxic because they contain those kind of LPS. Then okay, this is just about the ground staining stain reactions. So the magnesium, we already talked about it, right? So because like a gram-positive have a thicker heteroglycan, so then they can hold more the dye crystal violet. But once you add like a ethanol or some reagents, they so-called decolorize. So that actually make the cell wall shrink, right? So once they shrink, that means they will lose some dye. But for gram-positive, they actually can hold more dye. So that's why they can appear as the color, original color of the dye, which is purple. And then gram-negative, actually, they lose a lot of dye. So finally, it can appear as pink. Okay, do you want to like a little bit break, 2 to 3 minutes, and come back? So we still have a little bit of time. You can have some drink. So then go to the bathroom. Okay, we have like a 2 to 3 minute break. Okay, can we continue? Now you can see here just talk about a scenario about the osmotic like a response. So I found a cartoon online, probably just can help you understand better. Just to mention the two like a different situation. So one is so-called the hypertonic environment. I think that the so-called when we talk about the situation, actually talk about the salt concentration outside, right? Remember, so that means the salt concentration outside cells is less than what is inside. Okay, that's the hypo, means lower, right? Hypo, okay, I don't know what's the difference between these two. And hyper, so means higher, right? So that means that the salt concentration outside cells is higher or greater than inside. So if this is the kind of situation, then what will happen to the cells, like the bacterial cells? Here we just use the red blood cells. So they're actually probably basically the same. So then you can see if the salt concentration outside is the same as inside, so it won't change in cell, right? Cell kind of morphology. If it's lower outside than what is inside, then the water will come in, right? So make the cells larger. So so that's so-called hypotonic. So then you can see cell will swell, so finally will burst. So it's so-called lysis, right? So break apart, so lyse. And then if this situation is kind of opposite, so if I have a higher, like a salt concentration outside, then water will come out, right? So if we lose, cell will lose water. So then cause a kind of shrinkage of the cells. So this kind of phenomenon we call plasmolysis. Okay, then I really hope we at least know this kind of term. Okay, and when people talk about all the exams, plasmolysis, what it refers to. So that's basically kind of shrinkage or hypertonic kind of response, right? So then here just give a kind of experiment to confirm what kind of role like a cell wall plays. So if you remove those kind of wall, like a cell wall, so only remain kind of cell membrane plus cytoplasm and the organelles, right? We call those kind of cells called protoplasts. So basically just kind of cells without cell wall. Then we can put it like at the solution to see what will happen. So then this actually really happens, like a lysozyme, or if you put some antibiotics like penicillins, they actually can cause the inhibition of cell wall synthesis. So that's basically about it. So that's why peptide glycan is so important, because this is the target for a lot of kind of antibiotic drugs. So then if that's the case, if you put it like the water without a lot of salt, then that will happen. The water actually come in, right? We make the cells swell. So and then expand, right? So finally we break the cells, called lysis. This is just give me kind of evidence and show the cell are actually really important to protect the cells. Okay. Then we talk about the cytoplasm. So basically just kind of stuff within all the contents, right? Within the cell plasma membrane. This again, we talk about protoplasts. So what is a protoplast? Right? It's just a plasma membrane plus everything inside. And then cytoplasm we know, right? It's just a kind of most of actually water, right? And also contain like a nuclei, right? All the organelles. And this is just kind of very interesting cartoon I found online. So it probably can give you just a very strict like a comparison between gram-positive and negative. So you can see the gram-positive only have like a peptidoglycan as a cell wall, right? And again, so we just talk again and again because this is very important. So really, I want you to know what's the difference between these two kinds. Then you can see the peptidoglycan, then this is the outer membrane, right? Because once you know that, then you will know why, for example, later on, we talk about all other functions, like if you put antibiotics, so why there's a difference, right? So for these two kind of bacteria. So then you need to think about use of different antibiotics, right, to kill the pathogen. So that's why it's so important. Then the most important organelles, right, within the cell membrane. So one is the ribosomes. We know, for example, this is the cells. Okay, you can see actually there's a lot of the kind of plasm, the small dots, actually ribosomes. Again, bacteria need like a synthesized protein, right, very quickly once they want to grow. Also, we know the like E. coli, the cell, cell cycle, the life cycle, the whole life actually only 10 minutes. Think about it. So they need a synthesized protein very quickly if they want to grow very fast. Then for ribosome, we know this is just a kind of size or organelle or factory to produce a protein. If you separate into like the different component, we know they have two parts, right? This is just a different way to look at. So I have the so-called the small subunit or 30S subunit, or the large subunit, 50S subunit. So then once they combine together, just like two hand or sandwich put it together, right? So like that. So this is just a kind of a molecular structure. Then they will have a messenger RNA in between to produce a protein. So the S actually here is a stand for a kind of unit they call Sweetberg unit. And then you can see this S actually is basically is a kind of time called sedimentation rate. You can see this is seconds, 10 to minus 13 seconds. So because see they are so tiny, it's just very difficult to like quantify them or weight them, right? So then microbiologists or biochemistry, they find a way to quantify. Just use put them into, once you break cells, okay, put it into like those kind of centrifuge tubes, tiny one. So put in those high-speed centrifuges, so like the ultra centrifuge. So to centrifugate them. So in that way, to see how fast they can settle down on the bottom. So that's so-called sedimentation rate, okay? So here, remember, it's time, okay? It's not weight. So that's why, so we can see when we quantify them, for the separate, actually pretty easy to understand, right? One is a 30S, one is a 50S. So then once they put it together, actually say it's not like the mass, it's 80, this is actually 70. Oh, okay, this actually confused a lot of previous students, right? Why is now one added together is not 80S, 70S? Okay, because think about it, this is actually kind of speed, right? If you, for example, if you run by yourself with a small children, a kid, you definitely, if you have different speeds, right? Like a 100 meter, you want to compare, compete. But if you hold the children, the kids together, or to run, it doesn't need, doesn't mean you need the times put together, right? So you probably will be faster. Okay, I hope you can understand that. So that's just a kind of example. So that's actually something actually very important. Okay, that's one thing. Then that's some number actually I really hope we can remember. For example, for bacterial archaea, the whole part all put together is a 70S. So the size, in terms of size, okay? Then the small subunit is 30S, okay? Then most important, so for the RNA, we talk about over 16S, so which is also like the time or submission rate, okay, to quantify the size. The RNA is, the reason we call it 16S because is the sedimentation rate is 16S, which is a part of RNA within the small subunit, right? So the 16S, that's why we call 16S RNA or SSU RNA, because it only contain one RNA here. So when we talk about SSU RNA, that's the 16S RNA. Then 16S RNA is the way to quantify the size. Okay, then this actually is very important. Then is the 16S RNA, the size, which is come from small subunit. Okay, that's one. Then the total size of RNA, the ribosome is 70S for bacteria or archaea. But for eukaryotic cells, actually it's 80S. Okay, the total size. Okay, that's a different something I hope we can distinguish. For the ribosome size of eukaryotic cells, 80S. Okay, then for eukaryotic cells, they don't contain 16S RNA. They only contain 18S RNA. So that's the two differences. Okay, one is the total size, okay, between prokaryotic cells and eukaryotic cells. You can see they have a 70S. Okay, then for like a plant, animal, human, the ribosome size actually is 80S. Okay, then the small subunit RNA. So you can see only for prokaryotic cells 16S, eukaryotic cells 80S. And then you can see a little bit detail for them, the large subunit, small subunit for eukaryotic cells. You can see this is one is a 60S, one is a 40S. Doesn't matter. Okay, don't worry about it. But I want you to remember at least the difference, the total size. Once they add together the same, so it's now 100, right? It's 80. Okay, any question? This actually is very confusing for previous students. Okay, yeah, think about it. Then we talk about a little bit of nucleol. So then here, just give an example like E. coli. So you can see this is what it look like. So once you stain it, so you can see they actually occupy a lot of kind of space, right? But there's no like the membrane on it kind of, so it kind of mess. Once it release out, you can see they actually occupy a lot of kind of space. This is actually a so-called nucleus, nuclear or nucleosides, right? Nucleic acids or DNA, or we call this whole part called chromosome, right? DNA. Okay, that's basically the same. Then, you can see for bacteria, they have only one circular DNA, so double-strand DNA or chromosome DNA. Then, because they occupy a lot of space, right? They need to find a way actually to pack it, the so-called supercoiling. So we say that kind of nuclear proteins. I need to fold it. Except the chromosome DNA. So then you can see bacteria or archaea also have other those kind of DNA fall around in the cytoplasm. So also it's a kind of circular one, so small one, but compared with it, it's actually quite tiny. So we call those kind of DNA called plasmid DNA, which is very important. Okay, so the plasmid DNA actually is very important. You can see I have a lot of different kind of plasmid DNA. So we talk about more later on. So some like a plasmid, they actually can integrate into chromosome, so they can combine with chromosome together or separate from there. So we call episomes. Okay, they just a kind of different name. And then this kind of DNA actually they can is so-called inherit, so or they can reproduce. So it means that once the cell reproduce separates two, it still can keep it. It can reproduce another like a plasmid DNA. So in terms of the genes, so you can see all the gene in plasmid is so-called the non-essential. Means even the cells lose them, it won't kill them. So so-called non-essential is just won't affect the central metabolic pathway. Okay, they actually can still survive. So some gene like the antibiotic resistant genes just not involved in central metabolic pathways. And there's a different kinds, so we will talk about later on. So it's not, like you can see this here give an example, like a conjugated plasmid, resistant plasmid. This is so-called colicin, a kind of antibiotic plasmid, virulence, metabolic kind of plasmid. We know the like the lot of kind of professor for working on molecular biology. They actually have a lot of kind of plasmid. We actually can use them to help us doing a lot of kind of work. Then we probably stop here. So then in terms of the lab, so we only have a kind of online, right? Online labs this week. So I will send you, actually the already list on the Zoom link. So you can use that link. Then we will have a kind of Zoom kind of class. Okay, we talk about it like overall arrangement of this semester for the lab, and also talk about it like a syllabus and how to write lab report. And then my previous TA or my graduate student should make some video. You don't have to look at the video today, okay, this week. We probably just play in the class because we have a lot of time, right? I can play the video she make before. So then we talk about a lot of kind of lab safety. So those kind of issues. Then we will probably do the remain stuff because we cannot do any like a real like kind of lab, right? So we need to skip that. And then how to get it like a lab course we can do next week. And also remember to bring $15 cash. So I for the lab manual because I need to cover the basically like a printing wrap kind of cost. That's actually part of policy. So I need to get money back. What else? So okay, the safety issue. Okay, please wear mask. Otherwise, you cannot allow to go into the lab. We have two sections, right? So we talk about later more like in the lab section. Actually, it's a two, right? Like two to 450. So we'll talk about more. Okay, thank you. If you have a question, just stay here and talk to me.
Professor, I had one question about the large subunit of the ribosome.
Okay.
So it gave like two measurements, like one of them was like 5S and there was another one. So does that mean it's made up of two smaller subunits?
Okay, let me show you here. Okay, this one, right?
Yeah, is the large subunit just basically two small subunits together?
No, large subunit is just one piece.
Oh, so then what's the 5S?
Oh, that, okay, that's a good question. Okay, the 5S is the RNA. See, they have two kind of RNA here. Yeah, that part I forgot to mention. So for the largest subunit, they contain two kind of RNA. So
okay.
one is called 23S. Okay, probably see here. It didn't show here. Oh, it show here. It didn't show the picture, but probably this is better. So you can see this just like a kind of strand, right? So that's actually RNA. So we know the RNA is just like a piece of like a nucleic acids, right? they combine together. Then they have two kinds. One is 5S RNA. This is 23S RNA. So they actually embed in within the protein. We know the whole piece is actually protein, right? Then they have some RNA. So they actually kind of mixture. So then this is just a kind of a large subunit is just a kind of a piece of like a protein-based RNA. Then then the small subunit actually they only have one piece.
Okay.
See the gray part actually protein. Then they embed with the RNA. That's a good question. Yeah.
Okay. Thank you.
Okay.